Esteemed Chairman of Board of Trustees, esteemed Rector, distinguished academics, researchers, and participants. Welcome to the mini-symposium titled Future Directions in Space Science and Lunar Agriculture, hosted by the Faculty of Agricultural Sciences and Technologies. We are honored by your presence and thank you for joining us. This event highlights advancements in space research for human life and sustainable agricultural systems. Symposium aims to inspire innovative solutions for the future of the space-based life support systems. Before we start, I would like to briefly outline today's program. We will begin with the opening speeches. Following the opening, we will have a session chaired by the Dean of the Faculty of Agricultural Sciences and Technologies, Professor İsmail Türkan. In this session, we will hear from Professor Hideyuki Takahashi, Professor Atsushi Higashitani, and Professor Cengiz Toklu. After the lunch break, our session will start at 13:30 in room Y010. This session features Professor Higashitani, Dr. Higashitani, Dr. Şekerci, and Dr. Fuentes Merlos. They will discuss the effects of plasma treatments on seed germination and plant growth. Before we close, we would like to briefly talk about the entrepreneurship training and certification opportunities designed specifically for students and researchers in the life sciences. It is developed under the Deep Biotech project. It's a collaboration that we as Yaşar University are excited to be part of. We will give you all the details on how to get your micro-credentials and certificates through this project. Now I would like to invite our rector, Professor Levent Kandırlar, to the stage for his opening remarks.
Good morning, dear Chairman of the Board of Trustees, distinguished guests from Japan and Turkey, dear academicians and students, and guests. On behalf of Yaşar University, I would like to express our sincere welcome to you all to the mini-symposium on future directions in space science and lunar agriculture. After 50 years since the first landing on the moon by Apollo 11 mission, there is a gap of half of a century. And nowadays, human beings, they're attracted to go to moon, even making plans to settle down there on the moon or on a satellite on the orbit of the moon to give rise to maybe a space flight towards to the Mars. And all of a sudden, why after 50 years? And the recent development was April the 1st, Artemis 2 mission, Orion capsule, carried astronauts very near to the moon, and they have studied or gathered some data how a deep space flight or settling down on the moon or travel to the Mars will affect human body, mind, and well-being. It seems it's like 50 years, but we have an international space station up at the altitude of like 400 kilometers away. And for the last 25 years, distinguished scientists, they're making a lot of experiments there to study the effects on the human body, again, mind and the well-being. But all in all, besides these studies, it's important to solve one important problem if we are planning to settle down on the moon. How are we going to feed the colonists there? Of course, at the very beginning, they will be very earth-dependent, but later on, they must produce enough and healthy food to sustain the life there. How? Perhaps using regenerative life support systems, vertical farming, and tubes in which you're going to grow plants not just to feed the people there, but also the plants there will assist to produce oxygen as well as purifying the waste. It's not just the feeding anymore, solving all these three problems together. So in the International Space Station, for the last 25 years, there are a lot of experiments going on with these issues as well. In this mini symposium, let me start from our distinguished host, Ismail Türkan, Dean of Faculty of Agricultural Sciences and Technologies of Yaşar University. He has been studying plant growth under very stress conditions. Stress conditions because up on the moon, the settlers there, the colonists there, they must produce food under certain stress conditions, like high temperatures, like radiation, like soil toxicity, and microgravity. Ismail Hoca over there has been studying plant growth under stressors on the land like drought, salinity, and UV radiation, and microgravity, and so on. So actually, recall that Professor Türkan and the team from Asian University, they have designed an experiment that was conducted 2 years ago in the International Space Station by the first Turkish mission over there. In fact, it was one of the 13 experiments, and it was the first experiment. Professor Türkan, he has been collaborating with Japanese scientists, and two professors are with us today, and they have published a lot of scientific work together. And Professor Takahashi, who is the director of the Space Agriculture and Horticulture Research Center at Chiba University, and distinguished Professor Higashitani, who's a member of the Graduate School of Life Sciences of Tohoku University. And these esteemed researchers, they have been studying both plant growth and animal warfare under these stressor conditions, and they have conducted a number of experiments in the International Space Center, and they are affiliated with the Japanese Aerospace and Exploration Agency. So we are so glad to hear their cutting-edge research on these issues. My distinguished professor Cengiz Toklu is with us as well today, and he's a professor in the Department of Civil Engineering, Istanbul Aydın University. And he and his team is the first pioneering work in Turkey to produce the lunar soil, kind of a soil and dust, hocam, and just like exactly the same characteristics of the soil by the Apollo mission. It was 14, hocam. Okay. And he has a specimen, Turkish produced specimen, and he's willing to share with these soil structure and characteristics, and will apply our research there. So, on behalf of Yaşar University, we are indebted to these distinguished professors who kindly accepted our invitation and make this mini symposium to happen today. And we are really on the edge of having their cutting-edge results to hear from them. And my final comment is to enjoy the mini symposium in the morning session, and Dr. Higashitani, together with Professor Higashitani, she's going to share a cutting-edge research technique in the afternoon for the space students. So we are so indebted to have you all here, and enjoy. My best regards.
Thank you, Professor Kandiller. Now I invite the Dean of the Faculty of Agricultural Sciences and Technologies, Professor İsmail Turkan, to the stage.
Esteemed Board of Trust of Yaşar University, distinguished President of the University, distinguished esteemed scientists from Japan and from Turkey, my colleagues, dear students, we are really happy today since we were able to organize this meeting in a very short period of time by using a coincidence. The talk of my president was very informative and knowledgeable. So I'm not gonna go very deep talking about why space agriculture, growing plants in space, and why human being, because of its curiosity and ambitious urge them to go into deeper spaces, to the outer planets. What I would like to mention only is that about our work we performed with my Ege team, Barış Uzulday and Rengin Uzulday. If they show the slides, within several slides I will try to summarize what we have done in International Space Station when our experiment was brought into it by astronaut Alper Gezer Avcı. Why I am talking about this right now is that many people are asking me about the outcome. So maybe my talk will not cover all of the outcome, but in general will summarize what we have done. Okay, as you may recall that in 1923, Turkish Technical and Scientific Council and Turkish Space Agency opened a call informing that the first manned space flight will be realized to the International Space Station, and they asked proposals that Alper Gezer Avcı, astronaut Alper Gezer Avcı can perform it in the International Space Station. This one? Okay, so okay, and after the call, 13 experiment was selected by the committees, and it was one of our work called extremophyte. Why we put its name as extremophyte was that until our mission launched to International Space Station, there were no any study done on extremophytes that are adaptive to the very unusual environments on Earth. So it could be nice to see their responses under microgravity in space. So, as you can see from this slide that they visited our lab in Ege, when I was Ege University at that time. Now I'm acting as the Dean of Ege University. You can see that we teach them the methods that he will use in International Space Station. And yeah, Alper Gezer Avcı took this plant called Şirenkella parvula to go to International Space, ISS. So why we sent this plant? This plant is a kind of miracle. It has different very interesting and unusual adaptive traits, including it is, for instance, drought tolerant, it is salt tolerant, it is tolerant to lithium bore high temperature as well. So it's a plant that can withstand against multiple stressors factors which are relevant to the space environment. As Levent Hoca said, my rector said, that can be Moon or later on Mars. So to understand it is responses under microgravity to salt stress could be very informative to understand if it can sustain its adaptive traits under microgravity. That was not done before this experiment. So now we took our plants back. Alper, thank you very much for his efforts, and he brought our plants back. Now we are subjecting to advanced analysis, doing some molecular analysis. We are trying to understand how RNA expression under the space environment is regulated. It is rather important, and we will rate it with its tolerance to high salinity. And interestingly, until this experiment, no any halophytic or kysserohalophytic plant had been sent to the space experiment in ISS. So this is the first extremophyte that's sent into the space. Another aspect of this plant is that already American scientists sequenced its full genome. So we are now aware of its full genome, that we can easily relate the response and explain at molecular level. So why we thought with Barış and Rengin to do such kind of experiment in space? As my rector said, we have been studying for many years on plant stress physiology by looking at different adaptive mechanisms at physiological and molecular level, which are very relevant to the space environment. When I was in Tohoku University many years ago, I studied with Professor Hiroshi Suge, who was the founder of Life Science Unit in JAXA in Tokyo, which is now collaborating with NASA. We did not only do space experiment with Hiroshi Suge, but I learned many things from him, because also we studied water stress. So since then, we have been involving in doing different research in this field. And during our researches, we have been collaborating with my friends, Professor Dr. Hideyuki Takahashi, Atsushi Higashitani, and lately we met to Professor Dr. Aydın Cengiz Toklu from Aydın University. So on this occasion, we invited them, asked them if they could be informative about ongoing research related to Artemis and other spaceflight experiments. And they kindly accepted. And they are today with us. I don't want to go further to make you busy. I'm very sure that you are very impatient like me to listen what's going on in International Space Station and related to Artemis project. So, and within very short period of time, we organized, as I told you, this meeting. And during that time, we were very much encouraged by our chairman of Board of Trustees. Thank you very much for his support. And we were supported by our rector, Professor Dr. Levent Kandiller, vice rectors, my team in Faculty of Agricultural Sciences and Technologies, staff, all have helped a lot to organize this meeting. And for your participation, I would like to thank you all, and I hope that you will be benefited and you will be enjoying. Thank you very much.
Thank you, Professor Türkan. Before we begin with the presentations, I would like to share a brief biography of our distinguished speaker, Professor Atsushi Higashitani. After completing his doctoral program at Nagoya University Graduate School of Science, Dr. Higashitani began his academic career as an assistant professor at the National Institute of Genetics in Japan. Then he served as an associate professor at Tohoku University's Institute of Genetic Ecology, where he conducted significant research with within Professor Takahashi's laboratory. He assumed his current position as professor at the Graduate School of Life Sciences at Tohoku University in 2001. Since 2004, he has been leading pioneering spaceflight experiments using the Nemotet C. elegans to study stress response and aging in outer space. His previous leadership roles include Dean of the Graduate School of Sciences at Tohoku University, Special Advisor to the President, and Deputy Director for University Evaluation. Now I invite Professor Higashitani. He will present spaceflight experiments using the model organism C. elegans and health risk to the human body under microgravity in outer space.
Okay, so my name is Atsushi Higashitani. So thank you very much for our lecturer, the Reverend Kandari Sensei, and also the Ismail Tarkin Sensei. So just sorry. It is a great honor and great pleasure. That is also just very top university in one of the top most famous university attend and giving me such a great opportunity. Okay, and thank you very much. And next, so my name is okay. Can I use? Okay, this is better. Okay, thank you. Oh, this is my name is Atsushi. Sorry, Atsushi at meaning is maybe horse and she is a heart. It's okay in Turkish. Okay, I'm just graduate of the Nagoya University, just 1990, and then that moved to the National Institute of Genetics, assistant professor over the 7 years. And just after 7 years, we moved to the Tohoku area, Tohoku University. This 1997, so 7 years ago, just the Professor Ismail Sensei and Hideyuki Sensei and the godfather is Suge Hiroshi Sensei together to the same laboratory. After the, okay, and I'm very glad to introduce them. These 3 persons just really beginning of space plant science in Japan and Turkey established here. And the reason is so just that this period is just the starting of the space flight experiment, just using a space shuttle mission starting period. Unfortunately, that this period is that Japan has no opportunity using a space flight mission. Takashi Sensei is the first man, and later he present himself. Okay, but the 1996 is a Suge Sensei just retired period. He make one of the very interesting and important book prepared, Plant in Space Biology is a very cutting edge pioneer. And in this one of the section, so Suge Sensei write of the can plant normally and effectively produce seed in microgravity environment on space or not. This is his challenging idea, and also that this section. So he mentioned of the one of the 2 papers, Suge and Tsurukan Sensei, 1991, and the next one is so just Suge Sensei and 1991 paper cited, and this is also just the pioneer research, okay? And also nowadays, just that this is also just last March, we are celebrate of the Suge Sensei together in Tohoku University, and this period is also just this laboratory grow up such a level. And Professor Ismail Sensei just now is a Barry Sensei and Rengin Sensei together. And also the undergraduate student, Gunyuru-san and Akio-san and Keriman-san attend over the our Tohoku University, my laboratory. And both three obtained a PhD degree together. And now is Keriman-san come again to in my laboratory and working at the assistant professor position. And beyond the generation, just the increasing of this laboratory, so on. And today's afternoon, the Keriman talk about the current work, and she obtained the Dean Award in the PhD course. And also the Isabel-san, also she graduated my laboratory, and this is also just before prior to graduation ceremony. Just we had to stay over the one hotel together, and such a situation. Okay, so sorry, that very long introduction. And also this is also just afternoon, I would like to introduce of our cultivation in rice plant using of the Legoris stimulant using. And Legoris is this is not containing nitrogen source for plant growth, nothing. However, how do you supply with the nitrogen source? Well, our collaborator, Kaneko Sensei and Sasaki Sensei, established of the plasma technology produce of the dinitrogen pentaoxide, N2O5. It can be applicable, dissolved in water, that is also just supply of the nitrogen source on the lunar Legoris stimulant use. And still tiny, but they can grow up and make flowering. Okay, another project is just currently Japanese company. Dasai is a Japanese brewer sake company, just they are to the produce of the Japanese sake in ISS under the partial gravity condition. Now their sake, filtrated remain part, we are going to study of what kind of gene expression change and other aspect of yeast, what kind of change we will analyze together. Okay, our final dream is eating rice and drinking sake made on moon. Okay, so sorry that's so long a introduction. Okay, then just we would like to introduce my research activity. So just I use of the Nematoda C. elegans and mainly focus of the muscle alteration. Everybody, you know, the muscle is also just very important. Muscle and bone to keep up the standing of the strength and something so on. And then it is well known. Just muscle and bone is a well controlled and synthesis and also breakdown that is also well regulated. When you are exercise hard, bone and muscle mass are increasing. But oppositely, microgravity condition that is well known, bone and muscle atrophy accelerated in space microgravity. Yeah. And also not only microgravity, disused and bedridden condition, just the muscle and bone were shrinked. Similar way to the aging and also heat stroke and some of the genetically disease, dystrophy, and other mitochondrial disease, and also the cancer cachexia, that is the accelerator of the muscle and bone disruption. And astronauts have to harder exercise needed. However, this is one of the information data from JAXA. Just, you know, the arm bone, it's not so reduced per month, not so much change. However, lumbar bone and also the pelvis bone and foot bone, especially per month, 1.56% reduction occurred here. In addition, not only bone, just the muscle loss, about 10 to 20% loss per 6 months ISST, just average. And the maximum astronaut, maybe this astronaut not work hard, and then such a case is a 30% loss. Okay. And this muscle atrophy is maybe almost 10 times higher as compared to the normal healthy 60 years old. Moreover, the people just average is only 2% per year. And such a situation, it is well known, just weight on Earth were just gravity, Earth's gravity charged from the top to bottom holes. On the other hand, unloading condition in space, such a charged loading pressure disappeared, okay? And so this is also main issue, just the lack of weight, unloading condition. In addition, the microgravity effect of the each cellular level. Maybe that Professor Takashi talk about the plant, columnar cell and sorry, I forgot the name, but the inside stem cell and so. Okay, yes. And then these cell, the gravity response cell has a status. Okay, this is also just the sensor of gravity. And then in addition, recently to the mammalian culture cell, single cell also responds to microgravity. Maybe this case is a nuclei and mitochondria, maybe like a status function and keep up the position. Okay, now today my topics is not only two effects. So that is also just the tactile stimulation, contact stimulation, losing. Using Nematoda C. elegans, this is also very, very tiny animal, and the liquid culture, cultivation on ISS, and such a case, on the ground, so just the still precipitated slowly, bottom side, and hitting of the bottom membrane. On the other hand, the space microgravity cultivation, just levitation causes the floating situation continuously and not tightly hitting. Okay, this is the main, our focus today. And so you can see this is a Nematoda C. elegans. Inside the body has a lot of eggs. This is a hermaphrodite worms. And also the mature adult, their length is only 1 millimeter. On the other hand, the human case is here. And also the cell number is only 1,000. And the 300 neuron and 100 body or muscle cells. 600 is remain guard and other tissues. And lifespan is so firstly around 2 weeks is a maximum. And such a situation is very easily to analysis. And the egg to adult is only 4 days enough. Okay? And such a condition is very easy to analyze of the space mission. On the other hand, in the genome, it's very interesting. Such a tiny, however, more than around 20,000 genes, similar level of human, and 80% more higher homology. Dystrophy-related gene and Parkinson-related gene were highly conserved between human and Nematoda C. elegans. And then I have a very lucky to around 7 times of the space experiment carried out. First one is a use of the Soyuz capsule use, and 2034 is the international mission. And the next one is next to 3 JAXA oriented my PI experiment, and also the make a good friendship and continuously keep of the molecular muscle experiment organize of the ESA, my college teams. And this is also just the second mission of my first PI series. And this is, you can see that we use of the space shuttle Atlantis use. And we are sampling preparation after the launch. You are watching our collaborator together. And this analysis, we put on the baby C. elegans is the left side without food. And then the reach of the space, astronaut remove this U pin. And the right side is containing bacterial food for C. elegans. So baby C. elegans start of feeding, become adult, okay? And then the cultivation system in space, ISS, upper part is so just keep of the stand and the microgravity. Bottom side is a keep of the centrifuge, like a rotated and to the make a 1G artificial gravity. And to compare to the 1G and the microgravity condition, 4 days later, just baby C. elegans, moving behavior downlink to the astronaut Jeffrey to Tsukuba Control Center. And you can see that is also 4 days continuously grow of the microgravity. That is our swimming behavior, this one. On the other hand, the artificial 1G culture one, much more highly bending, much more strongly powered. Only 4 days, but the very big difference occurred. And then the frozen sample, just Nahoko, my partner and also collaborator, analyze of the protein mix and the gene expression level. She can find of the muscle protein and the cytoskeleton protein and the mitochondrial protein down regulation. Not only gene expression level and protein expression level, so on. In addition, just the body length, slightly but significantly reduce of body length, okay? And so, mitochondria, recently, nowadays, so it's just a lot of the NASA group, gene laboratory group to analyze of the astronaut and the mouse, all of the genome expression analysis. They have published of the 2020 cell paper, just focusing on the mitochondria inside of the energy power plant. This stress activated under microgravity condition. This is one of the very big issue. But still mechanism is unknown. We would like to challenge of the why mitochondrial stress activated under microgravity, okay? And also we can find not only mitochondria activity, but also we found of the decrease of the COMT4 gene expression continuously. The first experiment series that we found of the 25% and 33% expression level as compared to the housekeeping one of the gene here. Other the epigenetic experiment, we perform the 4th generation continuous culture in space. 4 day, 4 day, 4 day, astronaut exchange of the buffer and cultivated. And every case, that is around 30% and 50% level. And also the other UK group of the collaboration, 47%. And anyway, all of the case, COMT4 gene expression were down-regulated. COMT4 function is also dopamine inactivation enzyme. This inactivation enzyme reduction may be possibly original dopamine level reduced. No more need of the down regulation, probably. This is our hypothesis. Then we would like to challenge of the real dopamine level modulation. And the dopamine is also the commonly important, the consensusly important of the between Nematoda C. elegans and human. And this is important of the pleasure, cognition, memory, and learning, everything, activation, oh yes, motivation, so on. And such a very important neurotransmitter reduction rarely occur in space, microgravity. We challenge of the team, and Nate is also just our collaborator to together to NASA experiment. And then we try to the sample sharing and to just monitoring of the dopamine level by her. On ground, we are to use of the, this is a 3D crane rotation machine, randomly positioning. This is a simulated microgravity on ground. Under such a case, dopamine level is exactly reduced about 10%, so on. In addition, so just our collaboration spaceflight sample exactly reduce of the dopamine level grown in microgravity worms, okay? And so why dopamine reduce? One of the possibility is so just loss of contact stimuli, continuously floating condition, just affected or not. And then to confirm this hypothesis, we added of the 1 gram per cc white plastic beads during cultivation. And at such a cultivation period, 3D crane rotation, ground experiment, computational gene expression were recovered, and also the dopamine level also recovered. And also the moving, bending activity and moving frequency and body length is partially but recovered significantly. That's the meaning of the on-the-ground 3D rotation. However, contact stimulation is very important to keep up the higher dopamine level. And then the way to confirm of the muscle activity measurement on the ground experiment, 1G culture, just muscle, inside the muscle, calcium firing occurred of the bending site. On the other hand, the central picture is a simulated microgravity growing one. Muscle bending also lesser, and the muscle calcium firing signal reduced. On the other hand, apply of the beads, so just contact beads, strongly affect the strongly muscle power activated. Under such a case, is so just dopamine level recovered, okay? And to confirm of this ground-based experiment on space, so just we applied of the beads application, suppress of the microgravity real effect or not, to challenge, okay? And then this is so just 2 years ago, and 2004, 2022, we performed of the last my space experiment using Nematoda C. elegans. And you can see the white bag put over the 1 gram per cc plastic beads, okay? And then the launch of the Kennedy Space Center. And this time is we use of the SpaceX rocket to use. That is also much more lesser power as compared to the space shuttle flight, okay? Sound also very much more silent. Yeah. However, anyway, just slightly, okay, okay. We would like to introduce of the Kennedy Space Center, somebody to sleep, and then we'd like to introduce. And this is a Florida area, and the special, this center is a closed visitor center complex exists. You can see the Atlantis space shuttle watching of this visitor center, and this Atlantis of the my series experiment orbit, okay? And also this area is a this is a Banana River Center, and also the Indian River and something so on, and this is a Cocoa Beach. And the laboratories are very close of this Kennedy Space Center complex. And this is the inside laboratory picture. We are performing of the several time cultivation of the Nematoda C. elegans, almost 1 month. The reason is the space transportation system sometimes is delayed. The weather is not good and something happened. We are several times cultivated in the laboratory. In addition, this area is so just laboratory across of the parking area. It's still a lot of the nature exists. And then the Armadillo you can watching and Alligator is you can see. And that is a very good place. If you are challenged of the next time, please try and watching. And then my sample is so just the astronaut, Japanese astronaut Wakata-san is handling and the C. elegans cultivated and then the keeping together. And this cultivation system is which we use of the 2 types of bags. One is so just such a smaller bags without beads and with beads. And this analysis is so, firstly, we put over the fertilized eggs, only 30 eggs inside the bags. But these 30 eggs grow up after launch here, and then the adult become adult is 2 days later after launch. And then this adult continuously produce around 200 to 300 next generation egg. And then we monitor of this next generation egg become L4 and young fresh adult stage and watching of the behavior and frozen and sample analysis. Larger bag of the similar way, but the larger bag is we put of the L1 and L2 larval stage, each bag containing around 200 worms with GFP visualized image. And also the larger bag continuously cultivate of the additional 10 days old animal to monitoring of the aging effect. Okay. And then the firstly to the younger animal is what happened. And then this image is not much good. However, you can see the 1G ground control, central one is a microgravity, and left one is, right one is a beads application. So beads application is a much more tightly pushing of the beads, you can see. And the central one is a moving behavior is a much more weaker phenomena, reproducibility. But the motility cannot the measurement because the reason is the tightly fitting of the beads. And unfortunately, this analysis we cannot measurement. However, the body length is so just the exactly to reproducibility, reduce microgravity, and the beads application were recovered in space. And also the not only the in molecular level, just the C. elegans is so growth factor DBL1. This is one of the TGF-beta family protein. Such a protein gene expression level are reduced left side microgravity compared to 1G, and also the apply of the beads. Right side is a beads compared to microgravity, the completely opposite changing situation. Expression were down-regulated, microgravity, and the beads application were recovered. And also not only DBL1, but also the downstream signal factor, SMA1 gene and WALT gene were similar expression pattern appeared. In addition, we want to know why dopamine level reduced. And not only dopamine, after gene expression analysis, the other neurotransmitter, serotonin and something were also down-regulated. And then we focusing of the neurotransmission system, you know the synapse connection, okay? And then the pre-synapses and post-synapses connected each other, and also the lot of the dopamine and some of the neurotransmitter is spreaded. And such a case is active synapses. On the left side is inactive synapses. And inactive synapses condition, synaptobremin, this is a small vesicle, highly accumulated. This is also on the genetic mutant, dying mutant, the focusing of the nerve ring site. This is a brain of C. elegans. And then the, such a inactive, genetically inactive condition, you can see the lot of the strong signal accumulated. Okay, this is a resting stage. On the other hand, the wild type is signal is very faint. And then this technique use and apply of the space. And the result, you can see 1G condition, ground sample is a signal is a very faint and weaker signal, only 50%, 50% observation. On the other hand, microgravity condition, you can see the strong signal expressed one is 30%, and the rest 50% is a weaker but significant signal expressed. And the faint signal is only 10%. That's the meaning of the grow up in C. elegans in space. Just the neuronal synaptic activity were resting, reduced. And so apply of the beads, almost all observed sample is so just weak signal, reduced, and the signal was reduced, down-regulated. That means recovered of the neuronal activity. Yeah, and aged sample, we are focusing of the muscle activity mainly and neuronal network observation. Okay, it is nematode C. elegans, life, and day one adult. So we are hitting of the platinum wire, so just the go rushing and the punishment and escape. And the 6th day, until 6th day, just still highly moving escape. However, around 10 days and 50 days and just 80 days, just before deading situation, they cannot very grow, difficult to moving. And middle panel, you can see this is a C. elegans muscle mitochondria and muscle nucleus structure. Muscle mitochondria volume and very rich and highly connected. On the other hand, the aged one, 7 days and 10 days adult, the muscle mitochondria is a swelling start and the nuclear breakdown occurred, and then around 30 days adult, nucleus almost disappear and the muscle mitochondria shrink so much. Okay, this is a parallel connected. And also that this is you can see that we are to introduce of the calcium sensor. Just red color is a calcium. And the mitochondria age-dependent calcium accumulation occurred. And then such an effect is in over-accumulated calcium or cleavage by mitophagy function. And then the muscle volume loss and the swelling occurred. Okay? And then to monitor of the space from C. elegans. 4 days sample, just the 1G space condition, microgravity condition, and microgravity with beads, not so significantly different, nothing different. On the other hand, so just 10 days old animal, just the muscle mitochondria nucleus remain filamentous structure, upper one and the bottom one, another one. And on the other hand, you can see microgravity growing one, nuclear breakdown, and also the muscle mass, mitochondria abnormality and shrinkage occur. In addition of the beads, just recovered. That's the meaning of the number of population, just the severe muscle damage mitochondria, almost around 50% appeared. Okay, and the calcium level also increasing of the microgravity aged one. That's meaning of the microgravity condition. Aging was accelerated. And the red color is so just a stain of the pharogen muscle protein. And then the muscle protein fiber is not so much different. That's meaning of the mitochondria and the nuclei is a prior dysfunction occurred before protein degradation. In addition, we focusing of the motor neuron network. Just the younger stage is a motor neuron axon is a so highly connected dorsoventral and with like step image. On the other hand, 10 days aged worm sometimes is a so just axon brave, we call of the brave bubble appeared, and sometimes they are disconnected, but microgravity one, much more severe increasing of brave and much more breakdown, accelerated changes one. On the other hand, microgravity with bees, significantly phenomena were suppressed. And that's meaning of the not only muscle breakdown, but also the neuronal, motor neuronal breakdown also accelerated in space growth one. Okay? And then this is also just please take home message, summarize. And that is also just our one of the finding. Microgravity is a loss of contact stimuli, mechanical stimuli. And the long-term mission from baby to adult, still younger stage, neurotransmitter signaling were downregulated, resting condition. We cannot working, we cannot wake up and such a situation. And also the reduce of the motor neuron firing and body wall muscle firing, and also the growth factor replaced. Finally, body length was shrunk. And so this is a really occur on the ground in human. So recently, psychologists mentioned, one of the psychologists, the baby juvenile stage, mother and father sometimes are touching and infant massage, maybe cat and dog similar way, just that they feel pleasure. And such a pleasure feeling is a maybe activate of the growth factor, also the oxytocin, and also opioid and dopamine. Everything is activated. And during such a condition, brain development just well developed. Okay? And it's a very important issue. That if we think about in space, baby to grow up adult, the contact stimuli is a lesser condition. We should keep up such a risk exists, thinking about. In addition, so just our aged experiment, just the loss of contact stimuli is accelerator of the aging. This is one of the recently focusing. A lot of the soft massage is very important to prevent of the aging dental issue or something so on. And also such a may be bedridden patient. Also that we have to sometimes is also touching and sometimes is changing and such a maybe effective function. And also you can see I have my courage to perform of the mouse cultivated the gravity and the site of the use of the scientific report paper. And then you can see in microgravity mouse, that is also, sorry. Sorry, it's video, sorry. Sorry. Please go ahead. Next slide, please. Sorry. Okay, thank you. Okay, you can see the fecus is a clean up, and also uterus is just keeping. And then this is a Japanese made of the mouse cultivation system. Inside is a very clean keeping. However, this is a sleeping period and all of the untouched situation. On the other hand, the astronaut case, just they have to exercise necessarily every per week, 6 days, their performance of the around 1 hour exercise, several exercise training, okay? That's meaning of the exercise training may be effect of the contact stimuli occurred of the lot of part in ISS. However, mouse case is a very difficult to contact stimulation, okay? And however, this is my last message. How do you think in the bed? So we feel of the, this is a bed, just prior to sleeping condition. We feel of the relax and the pleasure. That's the meaning of the maybe dopamine and the oxytocin is produced. And then why we feel of the in the bed? That's the meaning of the maybe our back contact on the bed. On the other hand, astronaut, it's very difficult to sleeping condition, contact of bed. Maybe the longer duration of the space travel, it's maybe one of the another risk. We think so. Okay, thank you very much for your attention. And then finally, we would like to introduce a lot of them, my colleagues and something so on. Okay, thank you.
We will have the questions at the end.
Yes, of course, of course.
Thank you for your presentation, Professor Higashitani, Sensei. I now invite Professor Hiroyuki Tashaki. Let me introduce him before I invite him to the stage. After completing the doctoral program, After completing the doctoral program at Tohoku University Graduate School of Agricultural Sciences, Dr. Takahashi served a postdoctoral fellow at Wake Forest University in the US. Then, while working at Tohoku University's Institute for Agricultural Research, he also served as a visiting scholar at the University of North Carolina at Chapel Hill in the US. Upon retiring from the Graduate School of Life Sciences at Tohoku University, he assumed the position of the project professor at the Graduate School of Horticulture, Chiba University in 2023. He has participated in NASA programs and led 4 spaceflight experiments as the principal investigator. His previous roles include the Dean of Graduate School of Life Sciences at Tohoku University, President of the Japanese Society for Biological Sciences in Space, and representative of the Science Union for Human Planetary Habitation in Space. I now invite Professor Hiroyuki Takahashi from Chiba University to stage. His talk is titled Plant Space Flight Experiments and Future Lunar Agriculture.
Can you speak? Yes. Thank you very much for a kind introduction. And first of all, I'd like to thank the rector of the Ishar University, Professor Levin Kandila, and Professor Ismail Turkan for inviting me to this symposium. I am very pleased to be back to the Izmir again, and very honored to be here as a speaker at this symposium. And this morning, I have 2 topics. First, the spaceflight experiments with plants we have done. And second, our current approach to the food production and supply for humans' long-term stay in space. Yeah. Can you move? Yeah. Okay. The first topic is categorized as a gravitational and space biology. And the purpose of the gravitational and space biology is first, the use of unique environment such as weightlessness for obtaining biological knowledge. And this is very important because biological systems has adapted to the Earth's gravity for during their evolution. Okay? And second, the knowledge obtained from the spaceflight experiments should be utilized for establishing a life support system for human space exploration. And third, we need to apply such achievement for returning to our society on Earth to solve our various issues we have. Okay, I'm sorry, please go back. And in the background of this slide, you see the Japanese experiment module of a space station called Kibo. And please, next. And inside the Kibo module, there are lots of experimental instruments installed. And for plant experiments, Plant Experiment Unit, PEU, is available. So this has a plant growth chamber with LED light, camera, water supply, and ventilation systems. And this PEU is supposed to be placed in a CBF, as Professor Higashitani introduced. And the upper part provides a microgravity, and in the lower part, the plant is supposed to be exposed to the various G, including 1G, for comparison. So we can compare the effect of microgravity with artificial 1G at the same time in space. Okay, next one, please. Okay, you know the plants grow roots in the soil for obtaining water and nutrient. And plant shoots grow upward to obtain the light and carbon dioxide for photosynthesis. And this directional growth is called gravitropism, as you know, which is very important for plant survival, production, and ecosystem. And here is a rice mutant and barley mutant, which is impaired in gravitropism. And as Professor Higashitani introduced, when Professor Ismail Turkan was staying at our laboratory many, many years ago, he studied and characterized this Serpentina mutant and published papers co-authored with our boss, Professor Hiroshi Suge. Okay, so, all right, and this is the next example of the mutant we are using. This is a Japanese morning glory. And here the wild type grows up in response to gravity, but this mutant doesn't grow up. Just stay there, or if it grows, it just bend down. So we found this weeping mutant does not bend upward because they do not differentiate gravity-sensing cells normally. So as Professor Higashitani introduced, you see here the gravity-sensing cell, which has a sediment amyloplast on the bottom, surrounding the vascular tissue like this, okay? But in the mutant, you see no such a developed gravity sensing cells here. So we have identified the genes responsible for this mutation, right? So next slide, please. Okay, how about the gravitropism in the root? Okay, in the case of root, if they are placed in a horizontal position, they grow down, and the gravity sensing cells in the root gravitropism is columnar cells in the root tip. In here, you see the amyloplast sediment on the bottom side like this. This is the first step for the gravitropic response. And in the elongation region, there is a differential growth. That means a bending in here. So why this happens? So plant hormone, auxin, plays a crucial role, okay? And auxin has a polarity for its transport. And they come from the shoot apex through the vascular tissue to the gravity sensing cells, because auxin efflux carrier is localized on the distal cell membrane like this. So that's why auxin can move toward this way, all right? And auxin accumulated in the root tip goes down because in the gravity sensing cell, there's another auxin efflux carrier which localized on the bottom side in response to the gravity. So that's why the auxin came to the root tip, goes down, and that auxin is further transported by another efflux protein, PIN2, because in the epidermal cells, this PIN2 is localized on the proximal region of the cell membrane. That's why auxin comes from the shoot apex to the root tip and then return to the elongation region. So by this means, auxin redistribution is established. The lower auxin on the upper part and concentrated high level of auxin on the bottom side, which causes inhibition of the cell elongation, cell expansion, causing a bending downward, right? So next one, please. Okay, I'm going to show you another flight experiment we have done. This was done by space shuttle, and you may know the cucurbit seedlings are like cucumber, melon, squash, pumpkins. They produce a protuberance called peg between root and the hypocotyl after germination, okay, like this. And this peg hold the lower seed coat like this, and then the hypocotyl grow upward because of the gravitropism. So by that means, the seedlings can come out from the hard seed coat like this. So, however, in a space, what happened? The peg developed on each side of the transition region. So these results means that, okay, transition region has a potential to develop a peg on both sides. But on the ground, on Earth, the peg formation on the upper side is suppressed in response to gravity because peg is not necessary to be developed on the upper side for removing the seed coat, okay? All right, next please. And we try to find out a mechanism for such a, how can I say, we say the negative control of the peg formation by gravity, and what we found was reduction of the auxin level on the top side here. And auxin accumulate on the bottom side. So auxin is necessary to form peg in the cucumber seedlings. So that's why the suppression of the peg formation occurs on the upper side. So we searched some candidate for the regulator for such an event and found out that auxin efflux carrier we cloned from the cucumber, CS-pin1, is responsible for this event, okay? Here is a cross-section of this region, and you see in a green color, this is a CS-pin1 protein, okay? In the microgravity, you see, oh, anyway, this pin protein is expressed and localized in the gravity-sensing cell endodermis. And you see the signal of this protein all around the membrane in the microgravity. But after 2 hours exposure to the artificial 1G in space, you see the protein gets localized on the bottom side, on the, see, like this. So that means in the gravity-stimulated conditions, auxin is moving from the top to the bottom. So this endodermis is functioning as a canal for auxin transport from top to the bottom, right? That's why the auxin on the top side gets decreased, which causes a suppression of the peg formation, okay? Next, please. All right, then what happened to the roots in a space-grown cucumber seedlings? So before I go to the results of the spaceflight experiments, I'd like to explain about the hydrotropism, just in brief. There's a description of about the hydrotropism in old literatures, but researchers are very skeptical about the root ability to sense moisture gradient and grow toward the moistened area in the soil. So I was a postdoc in the US and working for NASA space biology program. And one day I found a small bottle labeled agiotropum. So I knew agiotropum, which is a mutant impaired in the gravitropism. That's a P mutant. And I decided to use that mutant and started to propagate the seeds by cultivating this mutant. And I came across a very mysterious behavior of the roots of this mutant. Here, the wild type, you see no roots on the surface, right? Well, because roots grow down in the soil. But in this mutant, you see there are lots of roots in the white color comes out from the soil. But shortly, however, these roots return into the soil again. So we are wondering what caused this kind of root behavior. And we did a very simple experiments, in the dark and under the light, and with moisture gradient and without moisture gradient and so on. And we found out this happened due to the hydrotropic response. So growing toward the moisture gradient, wet area in the soil. So this means, okay, a root responds not only to the gravity, but also the moisture gradient. Here's a gravitropism and here's a hydrotropism. But on Earth, hydrotropic response is interfered by gravitropism. So see the cucumber seedlings in a space. Here is a ground control. You see the primary roots grow down and the lateral roots grow perpendicular to the main axis. So that means in a horizontal way. But see here in the microgravity, a number of lateral roots grow toward the wet seed holding substrate. So I thought, because of this finding we did, I thought in a space, hydrotropic response appeared in the absence of gravitropic interference. So, please, next. To verify that hypothesis, we did another 2 spaceflight experiments. One is hydrotropy and one is CSP. And here, the procedure for spaceflight experiments. The number of tools, chambers and the fixation kit and water and, in a syringe, they were launched by space shuttle. And the astronauts operated experiments for germination and for establishing a moisture gradient in the chamber. And we monitored seedlings in the chamber. And finally, the astronauts fixed the materials by chemical and stored them in a refrigerator until the return to the Earth. The next one, please. Okay, in this chamber, we developed a moisture gradient between a water holding plastic foam and the filter paper attached to the other side, which contains water or sodium chloride solution to make a moisture gradient stronger, okay? And please look here. So in a microgravity, you see microgravity on the upper picture. So you see that all roots are growing toward the wet substrate by hydrotropism. But under the artificial 1G, like this, okay? So even in the presence of moisture gradient, roots just grow along the gravitational direction. So these experiments verify our hypothesis. Okay, roots has an ability to show hydrotropism, but it is interfered by gravitropic response on Earth. So next one, please. Okay, so we are very confident, about the root ability of hydrotropism, and we want you to know the molecular mechanism for this new behavior of the root. So I'm not going to the details here, but that's why we established the experimental system for the hydrotropism in Arabidopsis roots. And successfully, we obtained a mutant impaired in hydrotropism and identified the genes essential for hydrotropism, first in the world, okay? And maybe, here. Then we produced overexpressor of MIS1 gene, which expresses a MIS1 gene about 250 to 300 times more compared to the wild type. So such a MIS1 expressor shows an enhanced hydrotropic response like this. This is a wild type, okay? And these plants were placed in an artificial soil with a moisture gradient like this. So more roots grows toward the water source in an overexpressor like this. And under the water-limited conditions, the overexpressor shows a better growth. So this is a very interesting and important finding because this MIS1 is conserved in the genome of land plant species. So that means land plant crops, including rice or so on, they have a MIS1 gene. So what we are doing right now is to develop Mizuan manipulated crops for water-saving cultivation in space or in a semi-arid area on Earth. I think this is a very important approach. Next slide, please. Okay, as I mentioned, for the gravitropism, the columnar cell as a gravity-sensing cell and oxygen transport plays a very important role for the gravitropic response. But for hydrotropism, root cap columnar cell is not necessary. And also, oxygen is not a major player for the hydrotropism. In hydrotropism case, Mizuan and abscisic acid, ABA, plays a role which functions in the elongation region of the roots. So this mechanism is very unique, we found. So here, I'd like to emphasize the gravitational and the space biology led to the discovery of such a biological function. Okay? That's a very important point, I think. Next one, please. Okay, now let's move on to the second topics, space farming. Okay? So now human beings are challenging to live on Mars or moons, and the project representing such a program is Artemis program, as you know, okay? And the international project will build a small space station on a lunar orbit, okay? And that is called LOPZ, okay? And this could be a platform for manned activity on the moon and also further manned exploration to the Mars. Okay, next one, please. And for such a living on the moon or Mars, so as the president, Professor Levine mentioned, we need to produce a food. And the food needs to be produced and supplied on site. It's impossible to carry from Earth to the moon or Mars, for, let's say, 100 to 1,000 people for many, many years. So food production is very important for achieving such a purpose. But what we need to consider is environment, which is quite different from the environment on Earth or low Earth orbit in the space station, okay, ISS. So, for example, it's very far from Earth, low gravity, cosmic ray, UV radiation, vacuum, extreme temperature, complex of these environment, and very limited resource, okay? So under such a condition, we need to grow plants for food production. And the next one, please. And for such a purpose, we are believing to adopt plant factory system with artificial light, at least for the beginning of the project, okay? And Chiba University decided to establish the research center for space agriculture and horticulture in 2023. And that is because Chiba University had cumulative achievements in the research of urban horticulture, plant factory, engineering, and space-related program. So we have 3 research divisions. One is for new functional plants for space. The second, those plants need to be cultivated with highly efficient plant production in space, okay? And the system needs to be zero emission, emission-free in a closed ecosystem. So these three research divisions are working together to achieve our goal, okay? The next one, please. And the first division is aiming at understanding the effect of the space environment on plant growth and developing new varieties or cultivars which is suitable for space environment, okay? And second division enables the plant production under the, let's say, low gravity and low pressure or such an environment. And that means environmental control is very important in this division. But also, not only such a control, they need to have a system for automation and remote control of the operation in space. There's no labors, well, right now, the astronauts are enjoying to grow plants in the ISS and eating. But for space colony, it's very difficult to do so. And the third division is to develop a technology for utilization of the organic materials, zero emission, and materials circulation in a closed system. For example, our group is trying to decompose, recycle non-edible part of the plants or leftovers, human waste, and plastic to regenerate the fertilizer from those materials and then use for the producing the agricultural product again in a plant factory, okay? Next one, please. Okay, I'm going to show you some of our approaches, okay? As Professor Higashitani showed some of this, and this is called clinostat for simulating microgravity and partial gravity in space, okay? Here the two axes are rotating independently, which create the environment for the plants to be microgravity. And in such a situation, you see here another additional turntable here, which is making a centrifugal force by rotation. So that means that depending on the speed of this table, you can get a partial G between 0G to 1G, including 0.17G on Mars. Okay? Next one, please. And here's some example of the result. Yeah, please click. Yeah, in another one. Yeah, you see here the two types of clinostat. And see, the cucumber seedling is grown on a clino-rotated, machine. You see here a peg develops on both sides like this, like you saw in a space-grown cucumber. So we can simulate like this, the experiments. And here, the peanuts. Do you know the peanuts? No? Nuts. They flower on the above ground like this. But after the flowering, gynophore grows down into the soil and then produce a pod like this. But this gynophore is above ground organ, but not root. But behavior is like root growing down. So we need to understand the mechanism for this one. And here, the clino-rotated peanut plants. You see the gynophore growing at random direction. So questions are here, how we move the hard seed coat from cucurbit seedlings in space, and how we navigate the directional growth of peanut gynophore in space to produce cucumber and peanuts to eat. Okay, next one, please. Okay, in space, probably not only microgravity or low gravity, the organism is supposed to be exposed to the harsh environment, such as cosmic ray and UV radiation and so on, okay? And here's some of experiments by our colleagues, Professor Jun Hidema, and he is a photobiologist working on the UVB, and his idea was probably a space environment could be very stressful, not singly by microgravity, but also a complex effects of microgravity and UVB radiation and so on. So here is the results of the clinostat experiments. You see the simulated microgravity reduces the growth of Arabidopsis and moss like this. So in the presence of UVB radiation, so UVB also damages the plant growth like this compared to the control one. But in the combination of the both stimulus, UVB and microgravity, there is some synergistic effect causing more damage to the plant. So he is wondering if this happens in a real space or not. And they did a spaceflight experiments in 2024 and 25, and they are now analyzing the results, which might be a very important information for developing space crops, which is tolerant to space stresses, something like that. Okay, next one, please. Okay, for efficient plant production in space, environmental control is absolutely necessary. Here is an example of photosynthesis and leaf temperature as affected by microgravity. We did an airplane parabolic flight like this, which gives us 1G, 2G, and micro G for 20 seconds. And you see here, the photosynthetic rate gets decreased during the microgravity conditions. And leaf temperature increases like this during the microgravity. So this happens because there's no convection. You know the convection, the air movement, is not absent in the microgravity. So that means it causes a dramatic reduction in the gas exchange. So you can imagine, to solve this issue, we need to create artificial wind, airflow. So next one, please. So our group is using this kind of plant factory simulating lunar conditions, including airflow situation, okay? And well, basically, you know, they adopt hydroponics, the vertical farming, like this, for cultivation of the rice, soybeans, tomato, lettuce, strawberry, and so on. Because Japanese Space Agency listed 8 crops and vegetables to cultivate in space from the point of view of nutrition and technological, point of view, because they need to grow in a plant factory, in an artificial, condition. Next one, please. Okay, another one is, okay, think about the lunar environment, okay? 0.17 G, but for the construction of the food production system on the moon, the air pressure inside the facility are supposed to be reduced. Okay? In the ISS, astronauts are living in a 1.0 atmosphere, like in here on Earth. But for various reasons, the air pressure is supposed to be reduced, let's say, to 0.5 to 0.2 atmosphere, something like that. And we have already shown that Arabidopsis roots grow and flower normally, even in a 0.2 atmosphere environment. But question is now, as I said, we need to think about the complex effect of low gravity and low pressure for plant production. So here we have just developed a new facility. This is a air pressure controlled plant growth chamber. And we placed the cryostat like this inside. So that means we can examine the effect of combined effect of low pressure and low gravity on plant growth. Okay, next one, please. So I have introduced the 3 research divisions working on functional plants for space, highly efficient plant production, and zero-emission technology. Then finally, we need to integrate all elemental factors as one food production system, which is capable of resource recycling and so on. Okay? That is very important. We are working together to demonstrate such a system on the ground in the future. And at the end of my talk, I'd like to insist this kind of approach is supposed to contribute not only for establishing a food production system in space, but also for our society to maintain or develop sustainable society or industries on Earth. That is a very important point. Okay, next one, please. So, finally, I'd like to thank many collaborators and funding agencies, and I'd like to express my special thanks to Professor Ismail Turkan, and he visited us many, many times, and we enjoyed being together. Thank you very much for being together and collaborating for many years. Okay? Thank you very much for your attention.
Yeah, please stay. Yes, thank you. Thank you. Thank you, Professor Takahashi. Now we will have a short question and answer session. I will invite Professor Higashitani to the stage, please. We can have maybe 5 questions, please, from the audience. Barış Hocam. Yes. Mikrofonu açabilir miyiz? Gezici mikrofonları. Açılmadı mı? Şu gezici mikrofonu açabiliyor muyuz? Üstünde bir şey var mı hocam? Altından açılacakmış ama bir bakabilir misiniz?
Okay, maybe I can ask loudly. So I have 2 questions. First of all, thank you both for your presentations. It was inspiring and great, advanced cutting-edge research. I have one question for Asu Sensei. So do you think we can or we should develop like smart clothes so when
Yeah, yes, exactly. The one of the today I have no time, but the NASA group also mentioned in human body the fluid, the blood and also the water go up and then in space. But on Earth, our floor is just go down, gravity, but the body fluid system is also go to up continuously. And such a case on the Earth, just all of the fluid to go to up and then make moon phase. And such a case is so NASA is thinking about over the bed with lower pressure and then the bed reading period in space just go down to float. And such are already established. But the next, Bari Sensei point is very important. After I wrote over some of the such possible new hypothesis mentioned, and then some of the TV news, US show ask us some of the massage and some of the system needed or not. And also the one of the astronaut, Japanese astronaut interview, he said of the, I feel some of the, how do you say, the silent image, and that's the meaning of the loss of contact stimuli, maybe affect of the brain activity possible. And then as they said, of the also very essential for the contact stimuli. And such an astronaut said, in his case, just tightly bundling is a much more good sleep. And they are put on the gum, how do you say, the spring use and tightly touching of the back on the bed. And it's very good sleep, she said. And just in future, such a machine and some of the massage, something may be needed. This is my answer. Thank you very much.
Yeah, thank you very much. Yeah, that's a very important point. And yeah, we are working on such a program. Let's say we need to increase the part of edible part. So by manipulating genes for cell wall metabolism and so on, we can probably get increased not only harvest index, but also edible part. And even we can utilize the roots for maybe not only for the eating, but also they can be used for pharmacological purpose or some other ways. And important thing is zero emission. That means that we need to recycle everything, all materials. So for that purpose, yeah, we need to breed the plants. And also, not only for such a purpose, we are focusing on new cultivars, which produces more healthy components, like GABA or polyphenol, or even vaccine plants we are developing. Yeah. So, but so far, NASA has space variety cultivars, which is dwarf for making, let's say, product, from unit area. They need to increase the productivity, having a vertical farm, and then the height could be shorter, so to fit such a. But what we are doing right now is not only for such a dwarfing things, but also for a variety of, let's say, for example, in the case of cucumber, it has a parthenocarpy characteristics, right? That kind of trait should be introduced to other fruit vegetables because fertilization in space is a problem somehow, yeah. So, right. So there are lots of traits we need to consider for breeding the new cultivars for space. Thank you very much.
One question at the back.
I'm Dr. Koltuksus from the Department of Computer Engineering. First of all, I would like to thank both of you professors for I've learned a lot. And my question goes to Professor Higashitani. You did say that you're just using the beads to mitigate the counter effects of the zero-G gravity or the gravity anyway. So what are those beads are made of?
Sorry, pardon, one more.
Oh, okay, again.
Okay.
Okay. Let me rephrase again.
Yeah.
You're using beads to mitigate the microgravity, right?
Yeah,
okay.
yeah.
So the question is, what are those beads are made of?
Beads made of.
Yeah,
okay.
the beads. Right.
Okay.
Thank
This
you.
is also just the we apply of the 1 gram per cc weight plastic beads, polystyrene used. And on the ground experiment, we added of the different weight beads used. Some of the zirconia, that is a very heavy beads used. And such a zirconia beads is a 3D Klein rotation system is a completely negative effect. Baby C. elegans cannot grow up. And maybe the hard retouching is not good, and maybe the soft touching is very important. And that is an important issue. The now condition is that I'm slightly to 10% beads volume were added. It's maybe still too strong contact stimulation. And then we have to determine the much more suitable condition, reduce of the beads number, and also you are right of the much more weight, lower beads use or not, or such a very important issue. Thank you very much. It's okay? Yeah.
Thank you for your questions. Thank you, speakers, for the sessions. Now we will have a short coffee break. Let's join us here back in 11:45. Thank you very much. Thank you. Sevgili misafirlerimiz, kahve arasını sonlandıralım. Sizleri salonda görmek istiyoruz. Teşekkür ederiz.
Saygıdeğer konuklar, program başlamak üzeredir. Salondaki yerlerinizi almanız önemli rica olunur. Saygıdeğer konuklar, program başlamak üzeredir. Salondaki yerlerinizi almanız önemli rica olunur.
Welcome back. I hope you enjoyed your coffee break. I will invite the next speaker, but please allow me to introduce him. Introduce Professor Cengiz Toklu from Istanbul Aydın University. Dr. Toklu obtained his bachelor's and Master of Science degrees in civil engineering from the Middle East Technical University of Turkey, and his doctorate from Université de Pierre et Marie Curie from Paris, France. In his professional life, he directed and supervised numerous giant projects in Turkey, including a pontoon bridge, a long-span suspension bridge, a light rail transportation system, and several motorways. In academic life, he taught in several universities, starting from the Middle East Technical University of Turkey, serving in many cases as department head or dean. Currently, he is affiliated to Istanbul Aydın University. His research interests include application of optimization techniques to engineering, space civil engineering, non-linear analysis of structures, and construction scheduling. He is the author of several books, chapters, and scientific articles. Dr. Toklu is the developer of the method Total Potential Optimization using meta-heuristic algorithms, which is shown to be more successful than classical methods, including the finite element method in analyzing non-linear structural systems under constrained structures, unstable structures, and structures with non-unique deformed shapes. Now I invite Professor Cengiz Toklu to the stage. He will speak on production of stimulants of lunar and Martian soils for various purposes.
Good morning. First of all, I have to say that I am very honored to be here. Thank you very much to the administration of Yaşar University for this invitation, and to Ismail Hoca, of course. We had two excellent presentations in the morning from our Japanese friends. Now I will talk about this product, which is simulant of a lunar soil. The thing is, I will try to be with my team. We will try to obtain from this simulant big amounts, and so that we will bring here to this university sufficient amounts of soils, lunar soil simulants, so that these types of experiments will be done for agriculture and other things. Now, if we want to talk about how we are going to space, of course, the first environment of humans was around caves, small habitats. But later, of course, area got bigger, so that our area increased, and we have arrived at a position that globalization is finished, almost. That means that we know that now we are on a world. We know almost every corner of it. We know its resources. We know its nature, and its resources and nature defines our economy and technology. So we are living in this world. But now we are in a stage that we are moving in the space. We are conquering another area, and that area with new resources and with new nature will change us completely. This is a very well-known citation. The earth is the cradle of mankind, but one cannot expect to remain forever in the cradle, and we are at that point. Now, in 2050, we will be on moon, and our world will be world, moon, and the area between those two. In 2100, there will be Mars added to this world of ours. It is interesting that many friends here, many young friends here, will see these things. They will see that, the humans will be on Mars also in those years. And this graph, very simple, but shows how humanity changed its way of living. Before 2000, we were on the earth, and if you look at the elevation in logarithms, kilometers, you see that we are moving, very fast into the space. And this is another motto: man goes to everywhere he can go, and that is what it is being done. Now, in this new world, our first stop is moon. Moon is, Earth's largest natural satellite, formed when a Mars-sized planet collided with Earth 4.6 billion years ago. So that planet, Theia, was absorbed into Earth, and the rest of the debris clumped together to form the moon. So they got together. Therefore, the soil, on moon and on Mars, there are very much similarities. But the nature is very different. The gravitation is 1/6, no atmosphere, no magnetic field, the same side always facing the Earth. The lunar date is different. Temperature differences are very high as compared to Earth. There's radiation, and there is the danger of micrometeorites always. So these are the things that we have to consider when we are making some structures there. And in that new world, moon, water is very scarce. Actually, at the beginning, everybody was thinking that there was no water on moon, but later we have found that the research have shown that there is enough water on moon, especially on South Pole, in the form of ices, in regions which are called permanently shadowed regions, which are at the bases of craters. So that's why the humanity will, if it settles, it will settle, the first settlement areas will be the South Pole. There are moonquakes also. Since I am a civil engineer, these are important for us also. Now, the big differences are, of course, the natures are different, but also there are additional differences, difficulties of transport of manpower and resources, difficulty of maintenance, low efficiency of workmanship, and high degree of risk. Now, this study was done in 2010. You see that Professor Jablonski had considered 3 phases: phase 1, phase 2, phase 3, but this is now obsolete. Okay, we couldn't follow this presentation. Now we are, for example, at phase 2, there would be structures on the moon, but we are not at that point. Now, if this is the new consideration coming from our study, you see that in 2026 there are Artemis, but the first man on moon will be on 2027 next year with 2 astronauts, etc. That will continue. And for each activity, we have tried to find a year when that will happen. For example, for first agricultural trials, we have thought that it will start at 2035, and after 2040, there will be real agriculture on moon. So for all other activities, you can see that we will at the end have a moon city and a moon hotel, and at phase 3, there will be at least 10 to 50 people on the moon, and with no limit to the duration. But in phase 2, the duration will be 1 to 6 weeks. In phase 1, it will be 1 week. We are learning about moon and Mars soils. For moon soil, we have at hand also, some samples. For Mars, we have rovers which are sending us information from there. Now, the lunar soil, of course, now the problem, the thing is that we have 2 sources on moon, one of them is soil, the other one is water. So we have to before going there, we have to learn about them very much so that we can use them when we go there. Now, the first thing with the lunar soil will be that we will make groundwork, that we will have to make excavation, etc. So these machines also has to be developed and sent there. And don't forget that the soil of the moon soil and the earth soil has the same origin, so the power that an excavator will use is the same on moon and on earth, but the gravity on moon being 1/6, the excavator will not be very efficient. Here on world, you see that the excavators are very heavy, very big machines. When you cannot send them to moon, and even if you send them, their weight is 1/6. So that work is very difficult. So we have to invent new machines for doing it. Now, these are the lunar soils brought to Earth. Apollo missions brought 382 kilograms of soil. China's 2 missions brought 3.67 kilograms, and Soviet Union with Lunas, no men, but with sons, brought to the world 0.3 kilograms of soil. So we have less than 400 kilograms of soil on Earth. Therefore, we need more of these in order to make experiments, in order to be able to use them. So so we have to make simulants of these soils so that we can use them. We learn how to use them. Now, these are the chemical compositions of soils of Apollo missions. For others also, we have tables like that. You see that silicium dioxide, titanium dioxide, etc. We have all these oxides and the percentages of these oxides. And the thing is, on Earth, as you know, the soil is different between points A and B. On Moon also, we have the same problem. If you have the soil from Apollo 11, it is very different from soil of Apollo 16. So that doesn't, making a simulant of lunar soil doesn't mean that you know everything about lunar soil. Now you see here is the graph of comparison of all those soils. You see that they are very different. At the abscissa, we have the chemical components. So they are different. And they are, for example, on mare areas, that is, the sea-like areas, they are similar to each other. In highlands, they are almost of the same type. And there are also contact areas, which are not highlands, which are not marelands. So the soils are different. Now, if we are making simulants here, we can have different characteristics. One of them is particle size and shapes. We may want to make a simulant such that the particle sizes and the shapes of the particles will be the same on Moon. Or we may want to make a soil such that the bearing capacity will be the same. Or chemical compositions will be the same, and particle size and distribution. We are at point 3. In our studies, we are trying to make a simulant with chemical composition similar to those on the Moon. And this has been an international project. Almost, in many countries, they wanted to make simulants so that they will work on these soils, for example, to learn how to make concrete on Moon. Our studies made Turkey the 10th country on the world. So before us, they were Australia, Canada, China, European Union, India, Japan, South Korea, and USA. Now we are the 10th country to make these. To do that, from different points of Turkey, we got samples, we measured their chemical compositions, and then here you see, for example, these are only some of the samples that we have taken. And these are the properties of those samples. You see that we are using the same oxides as the Apollo soils. And then we have made an optimization program so that we can obtain the simulant of every type of soil coming from Moon. So in this table, on top, for each, for example, at the top we have Apollo 11. The first line shows the Apollo 11 properties. The second line, the line under it, shows the properties of our soil that we have made. This is something that was not done before, so that we are now at a point that we can produce every kind of simulant for those. Here you see that is a closer look to this table. And we have made, of course, an optimization program. You see that if P's are the percentages of lunar soil, if Q's are the percentages of our simulant, then we are trying to make delta minimum according to this equation. We are using a Jaya algorithm for doing it, and it is an iterational process. So this is the way how it goes. As you see that at about 6,000 iterations, we have found the result. And so, as I said, we have been the 10th country to do this. Now, this is one of the best results that we have obtained. You see from the graph, the blue is the soil from Apollo. The simulant is shown by red lines, and you have a sample like that. Now, later, we have obtained new samples from different parts of Turkey, and you see that on the right, we have obtained almost the same as the lunar soil, so which is better than the one that was shown. Now, regolith we call it, lunar soil. What does regolith mean? Regolith means that, you know, there are meteorites which are, you know, coming with high speed, and so that the soil changes its properties, having some particles from the meteorites also. So that soil is called regolith. And for radiation and micrometeorites, it is a good protector. If you cover your building with about 2 meters of regolith, then that will make good protection. Now, the next step, of course, for us civil engineers is to make, for example, lunar construction materials. One of them is concrete. And there are different types of concretes which are studied with sulfur, polymer, geopolymers, bricks, etc. And this study is still continuing. Civil engineer, there are, you know, engineers on the world which are working on them. And I will say here that I am member of the Aerospace Division of the American Civil Engineering Association. So in that association also, we are working on all these concretes and also on structures. Here we are seeing some properties of these concretes. You see that, you know, for us civil engineers, compressive strength is important, flexural strength is important, tensile strength is important. So all of these properties are being measured. In this graph, here we have a comparison between tensile strength and compressive strength of the different types of concretes studied. We are also working on some of them. Now, with these concretes, of course, there will be structures made like this. This is the general consideration, considered, you know, almost in all, you can see in every document. But also, we are considering these types of structures. This is our proposition. We have, you know, for humans, that part which is 0.75 atmosphere pressure, and an outer part which is 0.5 atmosphere, which can be used for agricultural purposes or any other purposes. Now, lunar water, as we have said, it is very important, and we have enough water on moon in permanently shadowed regions, and they are still working on that. These are, you know, two, three notes about this water, and here are the sources of water which are found in. There are not only, you know, this ice water on the lunar poles, but there are also water which are there with the origin of comets, for solar wind, etc. So there is also volcanic outgassing. There is also some process called volcanics on moon also. There are earthquakes due to them. There are also waters with of that origin. Now, these are detected in many ways, and Here are these methods of detection of these waters. And these are the locations. You see that it is started from 1991, and then they are continuing to find the locations of lunar waters. As you can see here on South Pole, the blue color shows the water deposits, ice deposits, and we are intending to use them. And the amount is not enough. There is enough water on Moon for all purposes. Here you see the craters' names and the amounts of water in there. For example, in the first one, there is 11 million tons of water. Of course, it's an estimate, and that can be used. Now, of course, this water is not pure water. There will be purification, etc., for use of it, either for humans or in agricultural purposes. Now, we have just a paper which is going to be published, which is published below, as you can see. And let us give a few notes about this research. The countries that have been to the Moon now, until now, is USA 44 times, Soviet Union 22 times, China 10 times, Japan 6 times, India 3 times, Europe Union 2 times, and Turkey also is on the way. And this is our satellite, which will be sent to the Moon next year. Let us hope it is going to be finished. And it will do this. It will be launched from Earth, go to the Moon's orbit after a few, of course, tours around Earth. It will go to the orbit of Moon, and then it will make a, controlled hard shoot on the Moon. Now, in our studies, we have these projects that are completed, completed or going on. The one at the top is development of construction materials for the lunar environment, performance of lunar regolith, simulant-based geopolymer concrete under vacuum, thermal cycling, and radiation exposure. It is a TÜBİTAK project. And the three others are for obtaining lunar soil simulant. And these are the publications about lunar soil simulants. And these are the conference publications. And the next projects are, again, on lunar soil simulants, and another project together with China. There will be 2 rovers sent to the Moon by an organization together with Middle East Technical University and China. And for those rovers, for them making their experiments, we will provide them lunar soil simulant so that they will make their experiments on soil using our simulants. And this is also another motto that I like very much. We went to explore the Moon and in fact discovered the Earth. Our Chinese, our Japanese friends have told us that, everything is being changed there, Nothing is remaining the same. So that change will teach us many other things. And then we will use them in Mars also. Everything, is changed. We will learn Earth at the end of all these works. And it will be the benefits of international cooperation. There is no need to count them. Everyone of us can consider about that. Thank you very much. I hope that, we will bring here sufficient amount of simulants so that in this university and perhaps with our Japanese friends, there will be good research to be done. Thank you very much.
Thank you, Professor Toklu, for this inspiring speech. Now we can take a few questions to Professor Toklu. Mikrofonu olan arkadaşlarım, şurada bir soru var.
How can the cost of transporting elements like nitrogen and carbon from Earth, which are necessary for sustainable agricultural cycle on the Moon, be balanced with on-site sourcing? In short, is biologically activating lunar soil or completely soulless farming more effective?
That part related with biology is something out of my area. We will send our soil to Ismail Hoca and he will deal with that part.
Actually, lunar regoliths or soil are not available to raise plants without amendments. So it should be rehabilitated by adding some compounds. Then it will make it better for plant growth. As you said, maybe also some microbes can be added. We will see. First of all, we need to receive the soil, start to testing it, if plants can grow, and under which circumstances. That will be main factor for us to understand and go further.
Okay, biological. Okay, I will answer of the biological biological view. So just that is a very interesting question. And so Professor mentioned all of you just the how moon produce. Just origin is so just the the crash of Earth. It's right. And then the lot of moon soil derived of the similar to Earth. Lot of similarity. Okay. And then however the longer history on Earth, just the first grow up of the cyanobacteria. This bacteria to make photosynthesis and then to produce of the oxygen and something so on. And under such a longer history on Earth, however, on Moon, maybe not too many biological organisms. And this is just to keep of the ancient Earth situation. Under such a case, is very difficult to the organic compound. Okay, and then we should make such a starting period to reconstruct of the moon situation to current Earth condition. And then it's very important, challenging issue. And also that you are right, is one of the new nitrogen source is very poor in the Earth, moon soil. And then we should need of the nitrogen fixation process needed. On Earth, some of the cyanobacteria, nitrogen fixation nitrogen fixation activity has. Such a cyanobacteria not only produce of oxygen, also produce of the ammonia. And then it can be utilized of the plant. Also, another limitation factor, you are right, and the phosphate. Phosphate exists in both Earth and Moon regolith. However, still keep of the crystal structure, and it's very difficult to solubilize. So phosphate solubilization needed to easily uptake of the plant. This is also another issue. But on Earth, some of the fungi that is associated with the plant root, such a fungi is also just accelerate of the phosphate solubilization. And then now maybe lot of the organism, lot of the variety of organisms living on Earth, such a situation is a very good cycling is and then will grow up everything. However, we should challenge Hideki Sensei last topics mentioned. So some of the good cycling and good not only plant, animal, and also some of the bacteria habitation also very important issue and challenging issue. Okay, is
Thank
it
you.
answer? Okay, thank you.
Thank you.
Modifying them to be more adaptive to the space condition. Have you heard about genome editing? Genome editing is a kind of technique that you can modify plants for your benefits, to make it desirable, to make it more adaptable to the condition existing in space. Am I right? You are also, yeah, they are doing genome editing. Genome editing is because molecular genetic technique that is now widely used and cannot be considered as GMO. Do you understand?
Yes, I
GMO,
understand.
there are many opposition about debate about GMO. However, this technique in general is accepted as suitable, which cannot harm and damage human health and animal. That's why there are many things to do in science. We should work hard and be patient.
Thank you.
Thank you for this very fruitful discussions.
Thanks for your presentation, Professor Toklu. I am Özgür Kilit in Mechanical Department, Mechanical Engineering Department. I am thinking about the soil type or the physical properties. Me and my students are trying to design Mars rover, and is it possible to be able to try your soil to try our design?
Of course. I mean,
But you said
you know, we are working mostly on chemical composition. But for rovers, you know, there are other properties of soil which are important. If we work on that also, perhaps we may arrive at a solution, like particle size distribution, like, for example, for rovers, they are using other types of materials in order to find the same density.
how sticky and
Yes.
others.
Yes. But now, up to now, we are working on chemical composition. But if those things are added also, that may be, of course, a good study.
Okay,
Thank you.
Thank you.
Thank you for the questions. Let's ask our questions during the lunch because we are a bit behind the program. Thank you, Hocam. You may return to your seats. Now we will proceed with flag ceremony. First, I would like to. Pardon? Oturtmayalım dediniz. Tamam. Anlamadım, özür dilerim. Buyurun hocam, böyle alalım sizi. First, I would like to invite Chairman of Board of Trustees, Mr. Ahmet Yiğitbaşı, to the stage. Cengiz Hoca'ya mı verirsiniz önce?
Tabii hepsine vereyim.
Cengiz Hoca'ya. Aha. Cengiz Hoca'ya verelim. Tamam. Biraz daha bekleyin hocam. Now I invite our rector, Professor Levent Kandiller, to the stage. And I also invite our Dean, Faculty of Agricultural Sciences and Technologies, Professor İsmail Turkan. I also invite our esteemed speakers, Professors Higashitani, Professor Takahashi, to the stage, please. Genç sağınızdaki. Şunu sen al. Bekle, bekle.
Also take this opportunity to thank to Helin, Ebru, Alperen, Seher, for all of the team of our faculty. Gave a great effort helping me during the preparations. Thank you very much.
Now I will invite everyone to stage for a group photo. Yes. Buyurun, fotoğraf çekeceğiz hep beraber.